Good morning, everybody. Day 8. Look at all the excitement in the chat. We made it. Thanks, Sophie. Getting everyone pumped up. I wish we had certificates for everyone. The endurance and the interest, the vigor. Welcome back to the final day. It's my pleasure and my privilege to get you started off. It occurs to me, I left my tea over there on the counter, so I'm going to grab that while we stew in the excitement of this lecture to come. Okay, better. I'm going to have liquid to talk. So good morning. Today we're going to talk about integrative research. In the morning we're going to talk about integrative research, multi-domain research, looking across data types, trying to give some next steps and some kind of ideas and a framework for how we might stitch together the different domains that we've heard about to some degree. Again, it's like a smorgasbord of research studies that I find kind of fit into various pockets of integrative research, different types, and a survey of some methods that you might also find useful. So one of the possible take-homes here is that you hear about a method or you see a research study or an approach that rings a bell in the science part of your brain and makes you think I should try this, I should learn about it, I should apply it to my studies. So we'll get started. I'm assuming everyone can hear me and everything's fine. Technical difficulties, schmechnical difficulties. No such thing anymore. Let's share the screen. Okay. And let's expand. Wonderful. So, welcome back. As I said, integrative research. I have the term whole person modeling here. I'll talk a little bit about that, but this is the idea. What parts of the person, aspects of a person, can we measure to get a more holistic version of the problem of solving problems in mental illness? We'll start with the land acknowledgement. CAMH is situated on lands that have been occupied by First Nations for millennia. These are lands rich in civilizations with knowledge of medicine, architecture, technology, and extensive trade routes throughout the Americas. In 1860, the site of CAMH appeared in the Colonial Records Office of British Crown as the council grounds of the Mississauga of the New Credit, as they were known at the time. And today, Toronto is covered by the Toronto Purchase, Treaty Number 13 of 1805 with the Mississaugas of the Credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. CAMH is committed to reconciliation. We will honor the land through programs and places that reflect and respect its heritage. We will embrace the healing traditions of the ancestors and weave them into our caring practices. We will create new relationships and partnerships with First Nations, Inuit, and Métis, and share the land and protect it for future generations. Again, the code of conduct. Everyone's been very nice. I'm not going to read through it. Keep in mind, in all aspects of life, let's keep a respectful environment going. And speaking of respect, language when we refer to people who suffer from mental illness and are affected in one way or another. Again, we've been kind of hammering this in. Just keep it in mind whenever you're talking about mental health or mental illness or psychiatry or neurology. The people come first, and we use that language throughout. And as I said back last week, don't shame yourself if you slip up. Don't shame others. Just remember it. Keep it front of mind and practice. Here we are at the end. You see, today the plan is me in the morning and group panel in the afternoon. And the group panel will be more of a Q&A, more of an open discussion. We have some questions lined up that have accrued over the course of the last 7 days. I really want everyone to think today, since it's not so instructive. I mean, there's some methods that we'll go through in a bit of detail, but you'll have room to think, kind of creatively and reflect on certain modalities or certain aspects of research. If you can think of questions that you'd like to ask anybody, anyone that you've heard from over the entire course, so they're all listed here. in any context, then save it. You can write it in the question box here. You can write it on a sheet of paper that's next to your computer. I think a lot of people still do that. I always still do that. I always have paper next to the computer. You might have a Google Doc open, a Word doc open. You might have something going on a Mac computer. I don't know what happens on Mac computers. So save the question. And in the afternoon, it will be answered. And it will be answered by very qualified, excellent people, experts, scientists, and that's how we will kind of build the afternoon session out. As I said, we're going to start with integrative research. There is a workshop this morning. So I'm going to spend the next hour and 20 minutes or so, up to 10:30, talking about research, showing examples of research and study designs, and I'm going to kind of end talking about a particular type of method that's designed to fuse high-dimensional data types. It's developed for molecular data, but can be integrated and kind of folded into a study design that includes other types of data, including neuroimaging data and electrophysiology data, demographic data, etc. So I will give an example of that, and in the workshop we will use it to actually generate patient subgroups. We're going to look for subtypes of individuals in a fictitious simulated data set that I've come up with, and look at some neat visualizations. So all of that is on the GitHub. You can follow along if you'd like. It's just an R Markdown script. There's a binder that appears to be functional that's in the Docker. Everything you need is in the RStudio Docker, and I'll go through that in the second session, so that you can have a resource and some code and some examples to go through on your own time if you'd like to sub in some of your own data, and kind of learn from that. And the afternoon, as I said, will be more free form, and we'll get to that. There are a lot of TAs today. I think I even missed a couple of them, which is interesting because it's not a very technically heavy day, and it's the last day. So I'm assuming by now most of you who are planning to follow along with the code will have had some of your technical issues sorted, but that certainly may not be the case. Anyway, if you recognize any of these names, you can reach out to them, call them out by name in the chat, make fun of them respectfully, jeer, poke jabs, make scientific puns. Post questions as well. Yes, on the Slack group, we have a Slack channel called Final Day Questions, which is another place on top of your paper or your Google Doc you can record general questions. But if you have questions about anything technical or anything scientific, these are people that you can ask about today's material. So without further ado, let's get into integrative methods, and let's start with some nomenclature and just overview bits. So what exactly is integrative research? And this is really just a summary of buzzwords. They do have meanings. We're not going to jump into all of them. Some of them have more specific meanings than others. The whole idea of integrative research is it could apply to a lot of different things. It could apply to an instance where you're looking at multiple different data sets of all the same data type. It could apply to identical analysis in structure that you're running in two different populations, right? It could be, that could count as integrative. It could be just a collaboration between two research groups. Who knows? But you'll hear words like multivariate, pluralistic, multimodal. Multimodal, of course, could refer to multimodal neuroimaging, and you might think neuroimaging is just one kind of type of data. Of course, these are arbitrary guidelines. Cross-disciplinary, who's to say which discipline is separate from another. Cross-species is a little easier. Biopsychosocial, also integrative. Model including biological, psychological, social determinants of health in one model. Collaborative. Network-based. I don't know. There's lots of possibilities here. Occurs to me that this might not be the most up-to-date version of these slides, but I think we're totally fine. I'll complain about it later if I make a mistake. Again, looking back, the first instance, if you look for integrative research, it's funny, modern medical research is pretty, it's quite recent, in human history. So you go back, published journals, you had Carl Pearson, who's the man behind Pearson correlations, a kind of statistical figure, did the first kind of widely received, the first integrative research method looking at both within group and pooled effects. It's like a stratified analysis looking at the effect of enteric fever inoculation in this was like overseas. This was wartime. This was like people were getting enteric fever, and when you inoculated them, there was reports of depression. There were like effects on mood and things like this, and he did like a stratified analysis. And then there are other kind of, much more recently, the development of meta-analysis, the idea of doing like second-order statistics and analyses of analyses. This is kind of the root of integrative research. And then actual integrative data analysis as like a formal concept, which was like introduced as the analysis of a single data set that consists of two or more separate samples that have been pooled into one. Quite non-specific and is more along the lines of meta-analysis than multiple data, kind of data domain bringing together, which is a newer concept. This is in the 80s. And by newer concept, I mean more along the lines of the terminology, what you hear of like biopsychosocial or cross-disciplinary or cross-domain, ideas that encapsulate very different areas of investigation. So what I mean by that is very traditionally you have camps of the conceptualization of mental health, and I guess more specifically abnormal mental health or mental illness, where many people believe that they take a reductionist, almost this hard reductionist perspective, where you can break everything down, everything we observe, all of your behaviors and delusions and anything that goes into a diagnosis of mental illness can be at some level or another, whether or not we're capable of measuring it yet or not, broken down into fundamental biology. It stems from biology. We can reconstruct it from biology. On the other side, again, traditionally, you've had people who say there is no way to actually do that. It's too complex. It's an emergent behavior of a fairly chaotic, unpredictable system that involves social and external these exogenous factors. And so there's no point in trying to be reductionist about it. You're always going to miss the mark in some way or another. Of course, those don't really need to exist at odds with each other unless you take the hard line. So, it's increasingly accepted that this is only really in the last 15 years or so that, models of mental illness will not hinge on a single modality or a single domain of data, like looking at biological or social constructs. We can't perfectly predict or understand major depression by asking people about recent events in their lives or deaths in families or stresses at work. That doesn't quite capture it. Just like you can't just take a blood test and tell someone if they're going to get depression. It seems self-evident now, but there was a time, especially in the emergence of the neuroscience revolution and the genetics revolution, when many of the kind of more hardline reductionist scientists who formed silos, I mean, they're not the only ones who formed silos. Lots of people had their own silos, but they said, look, we're gonna find the bits that they reduce to. And that's going to be incredibly valuable because it'll give us an objective yardstick for everything. And it hasn't panned out that way, as we know now, from complex genetics and from neuroimaging and the complexity and differences in state and trait. And this is where the RDoC framework came into play. We heard a bit about RDoC on day one, kind of throwback, which is stands for Research Domain Criteria, which is like an effort from a couple hundred scientists. It's based in the U.S., so it's like NIH scientists who kind of come up with this framework. And it is just a framework. It's not a diagnostic system. It really has nothing to do with diagnoses. It's not related to the DSM-5. It's actually, and this is funny, because I always was thinking about RDoC. It makes sense as an idea. So you give this idea of RDoC. The idea is there are a few domains, right, that relate to mental health. And they've been selected because they're of particular relevance to mental illness. You have cognitive systems, negative valence, positive valence, systems for social processes, arousal and regulatory systems. These seem to be like really important domains of investigation. And each one of them has a component that spans the biopsychosocial spectrum. Right? There are aspects to cognition that involve genes and molecules and cells and circuits and physiology and behavior, depending on what camp you're in. you'll be studying them differently. So what the actual output of RDoc is just a matrix. You refer to it as the RDoc matrix. It always sounded abstract. It's literally a table like this. So you're taking these ideas, genes, molecules, cells, circuits, physiology. The last one is paradigms. Paradigms can literally be like EEG is a paradigm, or a particular task that you give during fMRI, or just a cognitive task, the NBAC test, right, for working memory. That's a paradigm. Like, your whole research can be studying a paradigm, or a measure of self-report, or a circuit or a cell type, and you can contribute to one of these squares in this matrix, and it's constantly being updated. Right? Every time you run a GWAS on something that's related to attention, or perception, or one of these kind of domains, or these constructs, or a specific aspect of a construct, you run a GWAS, you get new genes, new SNPs that are associated with it, and the RDoc kind of framework is updated, and you can literally just go online and look at this matrix and see what are the genes associated with this aspect of this construct, what are the cells associated with it, and there's a whole bunch of funding opportunities through the NIH to add to this matrix. The whole point is to build this into a reference database. It's like a reference set of metadata that points you in the right direction for different constructs and serves as a way to guide further research. It's really all it is. Just again, there's more nomenclature. We'll hear a lot about I'm not going to speak too much on it because I think it's not often a useful distinction. Univariate, bivariate, multivariate statistics, statistical teaching. You'll hear about the distinctions between these two things, and univariate analysis is you're describing one variable, right? You can do things. You can estimate a mean. You can estimate variance. You can describe it. You can perform a one-sample t-test, right? You can look to see if the average of your one variable is different from some theoretical average. It's fine. Bivariate, correlation, two-sample t-test. Almost everything we're going to talk about is a multivariate test. Any time that you're going to include in any type of model, it could be a very simple model. It can be like an ANOVA. An ANOVA model is really a bivariate model because you have a grouping variable and some continuous outcome, and COVA is the extension. Anytime you include a covariate, you're talking about a multivariate methodology. Linear regression is, fundamentally not necessarily a multivariate analysis, but is, what's the difference between a bivariate and multivariate? I mean, it's more than one, right? That's my definition of multi anyway. Not an important distinction there, so free your mind. Data types. We talk about different data types. Just keep the concept straight in your mind. Don't overthink it. Don't try to overcomplicate it. We can be referring to a different data source, i.e. like an RDoc paradigm, i.e. a different tool, a different a survey versus a blood test versus a EEG cap versus an MRI scanner versus a PET scanner. That's a different data source, different structures. Just it is what it is. You can have the same data source with a different structure. You can restructure data. You can move things around. Just get these kind of concepts in your head. Fairly straightforward. Multimodal neuroimaging. You hear about this a lot. Same data source. Actually quite similar data structure. Same data source kind of from the brain, but different way of looking at the brain. So is it multimodal? Yes. Is it multi-domain? That's really up to you. There's a lot of lines being drawn in the sand here. What I'm mostly going to talk about today are efforts to kind of go beyond different aspects of the same type of measurement. So rather than looking at studies that are integrating data across multiple modalities, in this case of imaging, right? Looking at diffusion tensor imaging on the right, getting some diffusion weighted map of the brain and doing, like we heard from John, either some type of tractography or some voxel-based measurement or some connectomics, and then some structural measurement of a T1 MRI, and then a PET image of molecular distributions of molecules in the brain. That is integrative in its own way. Of course it is. I'm going to talk about kind of a level above that, treating almost all neuroimaging as a single group. And the same can be said for other types of data. Questionnaire data, right? bit different. You can ask about a lot of different things, but it's still a mode of self-report, right? It's distinct from an objective biological measure or some type of standardized assessment where there's no way that your perception or memory of that issue is going to influence the result. Some key aspects of designing an integrative study, if you're thinking about merging things together, Abhi touched on this yesterday, which is really important. Setting up your study design before you are necessarily accessing the data. I'm not going to talk much about this. This is something we could talk about in the afternoon. Hypothesis-free versus hypothesis-driven research. That is a fuzzy distinction, but something that we can talk about a lot, and it did come up last year in our discussion. The importance of having some theoretical framework for what you're studying, because in the absence of that, you either can't design a meaningful study or you can't interpret the result. So at some point, in your study design, if you're interested in just looking and seeing what comes out of something, at some point you're going to have to couch it in what's been done. And we saw a little bit of this yesterday in my bit on research that's useful. Without a meaningful scan of the existing literature, and without understanding what's been done and what the theory is for, the thing that you're trying to assess, your key outcome, your key kind of area, you're going to be lost at one stage or another, right? Either before you start analyzing or once you have a result. So save yourself the trouble and do it up front. Understand the structure of your data. That's almost kind of in the exploratory stage. You have to work with data. You have to get used to looking at what file types look like. What do the actual numbers look like? How are they organized? Is it in a vector? Is it in a matrix? Is it in an array, a multidimensional array? Is it in one file? Is it in multiple files? That's why I like showing, like with the genetics, it's a spreadsheet. It's a flat, like two-dimensional spreadsheet of subjects and genes or SNPs. And in Srijoy's examples of single-cell RNA-seq, you suddenly have another dimension, right? Not only do you have genes and individuals, but you have cells. You have multiple observations per person. So you're dealing with multiple matrices. It's a hierarchical system. And imaging data is organized into volumes, right? At the raw level, at different stages of processing, you end up with data that look quite different. If you take the raw kind of standard information coming out of the scanner, at some things are happening, Fourier transforms and stuff are happening in the scanner, as far as I'm aware of. I'm not a physicist. I don't understand all of the kind of molecular or the MR physics and what's going on there. John can talk about this. Probably very fascinating. But taking the raw, radio frequency, the kind of the signals that are being put into the tissue and then being reflected back out. You have to apply all these transforms, get these DICOM images, they're called, which are just the individual slices, right? Every time you're looking at, like in a diffusion-weighted image, totally different gradient, right? And getting a different angle of diffusion, you have to put those together and collate them into a three-dimensional volume, and then maybe you're collapsing that further. At every stage, you're getting a different structure of data. And then when you're looking at your study design, you're thinking about what you're going to measure, what your data look like. You have to be able to identify the appropriate opportunities for analysis, right? Because at the end of the day, even multi-domain research, you cannot be an expert in every domain of what you're doing, but in order to integrate it, you have to understand what the link is between the outcome of interest, what you're trying to measure, what you're trying to model, what the structure of your data are, and then be able to look at those two things and say, okay, well, this is going to work if I study it with this type of model. I need a tree-based model. I need a hierarchical model. I can do this with multiple regression. I need something to collapse the dimensionality. I need something to, I need to figure out a way in which I can get a meaningful result with the structure of the data and the outcome. That's the opportunities for analysis is the skill set. That's primarily comes from statistical training and experience. And reading how others have approached similar problems. And the second important part, and I've bolded it because it's really important, and again kind of harkens back to what I was talking about yesterday, look for modes and understand modes of internal and external validation. Once you have a result in your initial data set, and your initial foray, and your initial attempt at a hypothesis, hypothesis test, or even just a description of the data, what can you do to lend some credence to that finding, either in the same data that you have, which is often done through permutation or bootstrapping or some type of cross-validation, or externally, which can also be done, with types of cross-validations and with, withholding data or having replication or understanding how you can give it that type of validation. Really important. Final bit on study design and approaches to this kind of integrative modeling is to keep in mind the distinction, which again is not a hard line, but can help inform what direction you want to go. Variable-centered or person-centered. Variable-centered research is thought of as usually more traditional and is dominant in not just social sciences, it's dominant in biomedical science. You're looking at the relationship between our variables in populations, right? We're associating our outcome of disease or just our diagnosis with X, Y, and Z. What imaging markers are associated with diagnosis, what genes are associated with diagnosis. A GWAS is a variable-centered approach. Almost everything, a lot of what we've been hearing about is variable-centered. Person-centered is where the things that you're actually interested in clustering or looking at relationships between are not the variables, they're the observations, they're the units of measurement, they're the people. So this can be done by looking at person similarity. How similar are people to each other? How different are people to each other? What trajectories do individuals take, and how are those trajectories similar to each other? And I'll be talking about both types of approaches today, and what we'll do in the workshop will be a person-centered approach. There is another category, person-specific, which has been coined. I'm not making all this up. The point here is that it actually is very similar to the distinction I was talking about yesterday between precision medicine and personalized medicine. So it's kind of like a we're not there yet type of thing, but can be done. You might remember that data set I talked about, Russ Poldrack, scanning himself over and over and over, dozens of times over the course of a year or something, and having hundreds of blood draws, doing fMRI scans like every morning or something. And so that is a kind of the level of detail of understanding. Or wearables. I mean, this is big again, digital health. If you have all individuals in your study are being monitored constantly, portable EEG on your head. If Elon Musk had his way, he would be able to measure everything about you all the time, and then maybe we'd have person-specific models. And actually there are some kind of person-specific modeling approaches out there, but they all rely on the detail of data that's being collected and the density of data that's being collected on a person. So it's kind of like almost dystopian future type stuff. And they exist on a spectrum between variable-centered, person-centered, and person-specific approaches. And one interesting thing to think about, which is more kind of philosophical, is that these approaches, they're on a spectrum. You don't actually get you don't really necessarily get different information from looking at how people are similar to each other and how symptoms are similar to each other. Because when you think about, the simplest example of a correlation between two variables, you have a data frame where you have two columns and you look at how they're correlated. That's a variable-centered approach. To turn it into a person-specific approach, you're only transposing that matrix. Suddenly your variables aren't variables, they're people, but you're looking at the correlation across variables. It could be across different domains of measurement. That is the same data from a different angle. It's about the approach to how you're analyzing it. And it turns out you can get different data, different results. You often do because of the nature of biology and human processes. But this type of approach is not always going to, depending on what you're measuring, it's not always going to yield a different result. There's something called an ergodic process, which is a circumstance where if you have one individual and you measure them over and over again, and they have variability associated with something. Let's say you measure my mood over the course of a year, and you measure it every second, and then you plot that out. You have all of this variability in terms of what mood I could possibly experience. If that variability in my personal mood over time was perfectly represented or perfectly representative of the variability that you'd see in the entire population of humans, right? And that you could learn about the distribution of mood from just looking at me 1,000,000 times over the course of a year, and you don't need to measure 1,000,000 people, right? Then that's an ergodic process. There's no distinction. Of course, we know that individuals do not exhibit the full range of biological and psychological variability that can be captured in a population. So almost always we're never going to see these methods producing the same conclusion, because you can't simply observe one person and learn everything there is to know about people, right? You have to look at across people. So, bridging data domains. This is where we're going to go for the next 45 minutes. Looking at examples of research where we are looking between, again, there are some lines being drawn in the sand, cognitive and psychosocial measurements, sociodemographic measurements, neuroimaging, biomarkers and genetics, biology, and what you might call not biology. You could call it environment if you're thinking about prediction. In fact, when we talk about traits and phenotype from an evolutionary perspective, there is only gene and environment. Phenotype, the thing that you are, the way that you exist and what you exhibit outwardly, is only the product of genes and environment. Environment is everything that isn't in your genome. So that's a distinction that is, again, just specific to that kind of evolutionary thinking and this, the idea of phenotype. But for our cases, we're interested in examples of research where we are integrating different traditional silos of measurement, and I'm going to give some examples of those. I do see, so I'm not ignoring you, I do see that there is a question in the Ask a Question box. I'm not going to read it because I want to zoom through, but also because I might save it for the afternoon, for question fodder for the afternoon. So I don't even know who it is who's asked it. I apologize. I feel like it might be Akshay because he asked 20 questions yesterday, which is great. Keep them coming, Akshay. If it's not Akshay, I apologize. We'll get to it. So here are the examples I'm going to go through. It's spend less than 10 minutes on each of these. Certainly less than 10 minutes. So imaging genetics, gene-environment interaction, mixed and latent group trajectory models, common machine learning models, and high-dimensional omics integration in patient subtyping. On their own, each one could be a week-long course. But I'm going to give you a couple examples of research in each area to put some grease on those gears in the brain. Imaging genetics. What is imaging genetics? Imaging genetics is, some would say, an almost dead field. Unfortunately, it was the field that I grew up in academically. Kind of started in the mid-late thousands and was based on this revolutionary idea that why are we measuring, the association of genes with behavior when there are so many intermediates, right? We've learned about this spectrum here is I love this figure. I love this figure because of this it's like a ribbon, some kind of animation that came out of Microsoft Paint in the mid-thousands, but it stood the test of time. I mean, this figure's probably been shown thousands and thousands of times. But anyways, it's this groundbreaking idea. Why are we looking at genes to behavior? Because we've learned over the course of the last week that there are many intermediates, right? Genes form proteins, which form cells, which form circuits, which forms the brain, which form cognitive networks, which lead to behavior and temperament, and ultimately could create circumstances for mental illness. So why not look closer? Let's look at genes associated with the systems, with brain imaging. I mean, at the time in the mid-thousands, our ability to measure cellular phenomena was very limited. Single cell RNA sequencing was not a thing. It was not ready for prime time. Even RNA sequencing in general wasn't really up to snuff. It was all microarray. Everything else was too expensive. And our imaging, as you can see by the quality of this brain image, the imaging wasn't where it is today. So it was, it was pretty interesting at the time to think. And it was one of the only studies I'm going to show that I was, well, not one of the only ones, but only one that I kind of care about. I shouldn't say care about. Has a real strong place in my heart. This is my first ever publication, and it was once looking at the effect of a SNP in a myelin-associated glycoprotein gene. Does this single SNP affect the volume of your brain in a certain brain regions? And we just looked at lobes. We looked at the parietal lobe, the temporal lobe, the frontal lobe, and the occipital lobe, and we looked into populations, controls, and individuals affected by schizophrenia. Integrative research. Right? This is classic imaging genetics. This is totally classic. Pick a SNP. It's a genetic association study at the end of the day. is there one SNP that is associated with this hypothesis? We picked it, right? We're like, all right, this is a gene that's involved in, it's a myelin-associated protein, so it's related to the process of myelination, which is the myelin sheath is this like fatty layer of cells. It's actually like oligodendrocytes create this like wrapped layer of insulation around axons in the brain to kind of insulate long-range connections and make them more efficient at signal transduction. So we think this SNP could be functional. It's been identified in previous literature. Let's see if it's associated with the volume of brain tissue. And because we know brain tissue volume is associated with risk for schizophrenia, maybe there's something we can learn about the mechanism of how the SNP functions. And we found an interaction. We found that the individuals with one copy of the G allele at this SNP had a higher left parietal volume if they were if they had a schizophrenia versus if they were in the healthy control group. So in the healthy control group, there was kind of a lowering effect. If you had the G allele of this myelin-associated glycoprotein gene, you had a lower volume of your left parietal lobe. But actually, if you had schizophrenia, then that G allele had the opposite effect. It actually increased. You had a higher volume, suggesting perhaps some protective mechanism here, or a compensatory network. There's a lot of talking about kind of compensation through structure. Of course, it's my first publication, and there are many holes we can poke all day long, but it was lots of fun. Here's an example of something a little more integrative. So we saw a gene and brain imaging, which was T1 imaging, just structural neuroimaging, and a clinical diagnosis. We were looking between schizophrenia and individuals with no mental illness diagnosed. Here's an example of Alzheimer's disease. Alzheimer's disease offers a lot of opportunities for study that aren't present in other diseases. Primarily, it's because of the age of diagnosis. So, you can be diagnosed with Alzheimer's, late onset or sporadic Alzheimer's disease after the age of 65, so around 65 and older. So that's a long time to live. There's a lot of environmental exposure. There's a lot of biological things that happen over the course of your life. A lot of opportunity to kind of assess trajectories of people and how they may have a high risk profile or a low risk profile, and look at how and when in their lifespan they are developing the first signs of cognitive impairment that lead to Alzheimer's disease. And there's this idea of cognitive reserve, right? There's this idea that for some reason, whether it be your genetics or something else, genetics is often used because it's causal. It's something that you're born with, so we can kind of measure it. There are other things. Maybe you've been really active, you play a lot of chess, or you have a high educational attainment or really good social networks. It may be then that despite the fact that your brain is being riddled with pathology that causes Alzheimer's disease, you don't actually exhibit symptoms. You are resilient to that. On the other side, you might only have a little bit of pathology but start showing signs of decline because you have the opposite of it. You have like a high risk profile, highly susceptible. So think about that kind of idea. How do you measure that? Right? How do we understand this resilience to pathology and aging? And so a study, quite an integrative study, wanted to model this, and the way they did it was to define a single variable, construct a variable from multiple kind of integrations of different observations. So they developed a multiple linear regression of cognition in a cross-sectional sample. So this is just individuals in late life, and they modeled cognition as it's just a multiple regression with some demographic variables, education, age, sex, a measure of general health in the Hishinsky scale, as well as some measurements coming from MRI. So this includes white matter hyperintensities, which can be a measure of vascular health in the brain. It can pick up on stroke. It can pick up on fluid leakage and kind of just deteriorating membrane integrity. And then cortical volume and hippocampal volume, which are, they measure this kind of neurodegeneration in Alzheimer's disease. So if you kind of think of this vector of predictors as brain health, perhaps, right? And so when you model cognition as a product of all of these things, that's a multidimensional space, you end up with a line. So if you think about this x-axis here as being all of those variables, so I'm representing the multidimensional space in 2 dimensions, but the integrated brain health score, right, that's composed of imaging and demographics and general wellness, and we're modeling cognition. Well, it's not a perfect explanation, right? The model is missing something. And what does that mean? Well, it means that there are some people who have a really high level of cognition in green, right, who have better cognition than the average, right? Remember, this is a regression line, a regression to the mean. It simply means that the average expected level of cognition at this brain health level should be where the blue line is, but these people have higher than expected. So they have a residual that indicates that. The residual from this model is that their dot is above the line. And so the same can be said for individuals who have dots below this, right? The imperfection in this predictive model gives us information on the individual level about where people land in terms of resilience versus susceptibility. They're doing much worse, these people, than they should be for this level of brain health, right? Most people who have a high brain health score have really high cognition. For some reason, these people have a really low cognitive score. So they could be said to lack resilience. People who are right on the line will have a residual around 0. And then they went a step further, so that's already fairly integrative. We have this measurement, but then they did a GWAS on residual cognition. So they termed those residuals from that model residual cognition. So any people, remember, those are just dots of individuals who have a high or low residual, have this kind of residual cognition, and put that as the outcome in a GWAS model with covariates across the whole genome. And in doing so, identified places on the genome that confer risk, right, susceptibility or protection against late life cognitive decline in the face of low brain health, right? So you have many kind of steps here to this integration, but they've brought together kind of self-report of health measurements of physical health, sociodemographics, biological sex, brain neuroimaging, and genetic background, all to make kind of mostly variable-centric conclusions about the relationship between genetics and susceptibility and resilience to dementia. Another area of integrative research that's pretty prototypical is gene-environment interaction, just by the fact that it kind of, in its nature, as I mentioned before, in evolutionary thinking, and it's kind of a fundamental way of thinking about who you are and what anything is in terms of living systems. You are a product of your gene and your environment. It's nature and nurture. So looking at how those two things interact is quite integrative, but covers a broad range of ideas. So to just be clear, there are a couple of ways to conceive of gene-environment interactions. The way that I find kind of easiest to grasp, you can think of a matrix like this, where you have a burden of exposure of some kind, some environmental exposure, okay? And you can have a low exposure or a high exposure. You can ignore these number values. They've just been given. The paper kind of goes on to formalize this. It's not necessary to understand it. Genetic burden can be thought of in the same way. Now, you have 3 groups here. This could very easily be 3 genotypes, right? Like a AA, AT, and TT genotypes at a particular SNP. Could also be a polygenic risk score, low polygenic risk score, middle, mean, and high polygenic risk score. You can think of it that way. And just where you fall on this as an individual dictates your phenotype, indicated by the palm outward, kind of strangely poised muscular figure in green and purple. So you can see here that your phenotype, the green or purpleness and size of you, is determined interactively by your exposure to environment and by genetic burden. Neither one alone will explain your phenotype. So if you look at a high genetic burden, some people with a high genetic burden will be small and purple, some people will be large and green, right? And that is clearly not related to your genetic burden, because they all have a high genetic burden. It depends on your environmental exposure, right? Same thing for environmental exposure. People who have a low environmental exposure are both large and green and small and purple, and that is not related to your environment, right? And that's determined by your genetic burden, high and low. So you need to know both of these things to identify where someone falls on the spectrum. Another way to think about it, similar idea, different publication, environmental exposure on X, trait value on Y. Both of these, A and C, are gene-environment interactions. So in this case, you can see person with genotype 1 has some trait value over environment. It changes over the course of environment. It changes differently over the environment than person with the second genotype. Right? Individuals with 2 genetic backgrounds have a slightly different response. In this case, a little more extreme. Right? If you look at this level of environmental exposure in the middle in panel C, 2 different genotypes have very different values of their trait. Right? 2 people who are raised in the same environment two people who both smoke, two people who have the same job, two people who share levels of stress at work, but two different genotypes have very different trait values. However, people in this environment, right, people with a really high level of environmental exposure to this have no variability in their trait, regardless of their genotype. And on this, you have totally normal reactions to environment that are the same. There's a genetic main effect here, right? At all levels of environmental exposure, your trait is going to be different if you have genotype 1 or 3 here, these two different genotypes. But the difference between them is the same, regardless of your level of exposure. So that's how we're going to think about gene-environment interaction. Here's an example of a gene-environment interaction. A study, again, this is in Alzheimer's disease. Our y-axis is cognition. It's actually a score of cognitive dysfunction. And so that means the higher value is worse cognition. And on X is one of the proteins that is thought to cause Alzheimer's disease. It's certainly a diagnostic requirement is this beta amyloid protein. And this is measured with neuroimaging. So you have here a cognitive score that's determined by an interview, a kind of a set of tasks, and then you have a molecular imaging modality here. And then we take a look at the relationship between these two things. The higher your amyloid, the worse your cognition. You can see that's a kind of general trend, and that makes sense. Worse pathology, worse cognition. But if you stratify the individuals, you group them according to genotype at a particular variant, you see that the relationship between these two variables is moderated, is different, depending on amyloid. So this is exactly like the curves we just saw on the right here. We have an environmental exposure, right? Some level of proteins that are building up in your brain, which can be thought of as an environment. It is a pathological environment in your brain. It's almost an exposure. And then our main outcome here, our trait, cognition. And that curve, these aren't curves, this is a linear model, those lines have different steepnesses. The effect is different depending on what genotype you have. A gene by environment interaction. There is some altering of the effect of these on the other. You can think of it as both the relationship between an exposure and trait modified by genotype. You can also think that the relationship between the trait and genotype is different at different levels of exposure. If you look at high amyloid individuals, people with the TT genotype have worse cognition than the C genotype. But at very low levels of amyloid, the pattern is the opposite. You can see the individuals with the TT genotype, on average, actually have lower cognition, or about the same cognition as the other genotype. I'm going to touch on the idea of dimensionality. When you have a lot of variables, as is the case in GWAS, as is the case in gene expression dataset, which is where this example is going to, using gene expression to infer environment, you have a lot of variables that you've measured, and that equals a curse. So, I probably should have taken this out because I digress and I'm going to run out of time. This is the black spot from Treasure Island. It's a curse. So it's kind of like a curse. This is Long John Silver is reading this. He got the black spot. He's been accused of a crime by the pirates, but this particular black spot, he's been convicted of a crime. This black spot was ripped out of a Bible, so it's just doubly bad. It's bad because he's been convicted of a crime in pirate language, and also was ripped out of a Bible, which is bad news. You have a lot of variables. It's thought of as a curse of dimensionality. It's because you have to deal with the possibility that A, you can overfit models because of random chance, and B, you have to kind of penalize yourself for looking at a lot of things statistically. We can chat about that in the afternoon if you'd like. One way we deal with dimensionality is to reframe original data points, and I'm not going to spend much time on this because it's worth looking into if you're not familiar with principal component analysis. You can just think of it as a way of taking original data points here, kind of exemplified by a y variable and an x variable. You can see there's a relationship between the two of them. Principal component analysis, it just identifies a transformation of your data, and it can't just be two variables. It could be hundreds, it could be thousands of variables. It transforms all of them in such a way that it creates new variables, principal component 1, principal component 2, all the way up to the number of variables you have. And each of these new dimensions, which is kind of a way of mapping your original data into some new space, it's called latent space. But the advantage is that the latent space has a mapping, a direct mapping to your original data points, so you don't lose any information. But your new variable coordinates, your new variables, are oriented in a way that they explain most of the variability, the linear variability and covariance in the data that you have. So if you have 100 perfectly correlated variables and you want to look at all 100 of them, that would be pretty silly. because they're all perfectly correlated with each other. You'd only have to look at one of those variables to understand the relationship with all the rest of those variables. If you ran principal components analysis on a data frame of 100 perfectly correlated variables, you would end up with one principal component that explains 100% of the variability in all your data. It collapses 100 variables into one variable. You would, in fact, get 99 other principal components. They would be useless. They would explain nothing. It would actually break down. But your first principal component would be one variable that explains all the variability. Of course, in data sets, naturally, there isn't perfect correlation between all of your variables, but it will capture as much of that correlation as possible. This is what we saw when I was showing ancestry determination in UK Biobank back on day 3, which is when we're trying to identify ancestry, large patterns of genetic similarity and differences between people in a broad population, we just perform principal component analysis on the raw SNP data from all these subjects. So this is taking a matrix of 400,000 people and 147,000 independent SNPs. So these are kind of pruned down from the list of millions. 147 totally independent, well, they aren't totally independent, independent loci because they're not in linkage disequilibrium on the same chromosomes, running a principal components analysis and projecting each person, each one of these dots is a person, into this latent space. And we can then see on this new kind of coordinate system how similar people are to each other when we've taken into account all of the variables in our data set. This is, in fact, a person-centered approach to analysis. And one of the ways in which we've gone about using this principle is to identify environment in the brain. In the previous study, we used PET imaging to do it. In this study, we did RNA sequencing of brain tissue. And we used, here it's called LV inference, latent variable inference. It's essentially just principal components analysis paired with another step of cellular embedding where we come up with latent variables, our principal components, that seem to be biologically plausible or representative of certain biological processes. And then perform a gene by environment interaction. So it's a fairly complicated workflow, it looks like, but it's really RNA sequencing, coming up with new variables that represent the environment of the brain, and then adding in genotype data. And then we're doing exactly what I described before, gene by environment interaction. Look, 3 genotypes, environmental exposure are latent variables, and gene expression. In this case, raw gene expression per variable is our actual outcome. The idea behind this is that genetic regulation in the brain, right? That remember that central dogma from genotype to gene expression to proteins is not always going to be a perfect kind of one-to-one-to-one process. In fact, it's most often not. There are a lot of things about the local environment of the brain that might change the relationship between genotype and gene expression, and the whole idea here was to kind of use this latent variable approach to understand those conditions and how they vary the effective genotype. So we end up with all these latent features, and we correlated these latent features with actual phenotype data from the participants, right? So we had RNA sequencing from hundreds of people. We also measured cognition, clinical diagnoses, personality traits, age, sex, all of this, lifestyle features. And you can see that these latent features actually map onto, some of them more strongly than others, a lot of these actual variables. So that there are some features, for example, that seem to quite strongly represent smoking or alcohol use, others that represent aspects of personality and kind of map onto those. And as an example, what we get, again, this is the same kind of idea, gene by environment interaction, a particular genotype here, the CC genotype, 2 copies of C. If you had 2 copies of C, you really had a very different regulation of this gene, SPADA7, under environmental constraints of a particular latent variable 71. So it's a little more abstract. It's still kind of in the omics area. I wanted to bring this up because it's an environmental approximation. We did correlate it with external environmental variables to kind of look at how the local environment. Remember, this is all about brain tissue. We're measuring RNA sequencing expression in brain tissue. So by using that expression data and this latent variable analysis, we're reconstructing the local environment at the level of where the gene expression is occurring. This was brought up briefly in a previous session. How do polygenic scores compare to each other? And this is an example of a study that did a kind of polygenic score by environment interaction. They've sorted here these polygenic scores for different traits on the left here, and this was all done in UK Biobank. They've sorted them by how well or how much the polygenic risk score added to the prediction of a baseline model that includes age, sex, and genetic ancestry. And these area under the curve models, again, a value of 0.5 is random chance, a value of 1 is perfect discrimination for cases and controls. And you can see depression ranks kind of near the bottom here. So depression added 0.014 to the AUC value of a model of just age, sex, and genetic ancestry when actually applied to this data set. So this is something that you can come back and look at this and look at the actual paper to look at these in more detail. It's a nice kind of a nice explanation and a unified framework for looking at the performance of different polygenic scores in the same sample. For this talk, it's important to point out they went further to look at the stratified analysis, gene by environment interaction, the incidence rate of the diagnosis of interest. In the top left here is depression, right? How many people in the sample had depression who also had a low, intermediate, or high value of the polygenic score, and then walked a certain amount per day, right? Minutes of walking, less than 20 minutes, 20 to 60, 80 minutes, I guess, and more than 80 minutes. So this is gene-environment interaction, textbook, right? If you had a high polygenic risk score, right, the takeaway here, a high polygenic risk score meant that the effect of walking a lot was much more pronounced, right? If you walked a lot and had a high genetic risk for depression, then actually the incidence of people who had depression in that strata was the same as if you had a low or intermediate polygenic score. It almost nullifies the effect of genetic risk. Whereas if you didn't walk, if you were stagnant and you had high genetic risk, you had a much higher incidence of depression. I really like the way they did this because they put incidence rate on the y-axis, which you don't often see because it kind of reveals to you the true meaning, back to clinical utility. What's the scale on which we're measuring this? What's the actual difference in terms of the risk that you have, the kind of the risk ratio, not just odds of being in one group or another. So truly only 0.1, the incidence is still quite low, but it is different. This is also a great opportunity where I get to highlight some of the excellent work of colleagues in the centers. This is work led by Michael Weinberg, who's a postdoc with Sri Joy Tripathi, and they published a paper earlier this year that received some attention, and it's a great example of gene-environment interaction because they calculated a schizophrenia polygenic risk score and looked at the interaction between polygenic risk score and cannabis use. And basically it turns out that depending on your and so it's the interaction of cannabis use and polygenic risk for schizophrenia in terms of how it kind of affects hallucinations, delusions, different types of delusions, and psychotic experiences. And basically it comes down to the fact that depending on your percentile of polygenic risk score, the odds of, if you smoke marijuana, the odds of being, of experiencing persecutory delusions or delusions of reference can be quite different. It can be quite a dramatic difference in terms of your odds of developing these delusions, depending on your genetic profile. So it's an integration of a lifestyle factor of smoking, of smoking, and then also of schizophrenia genetic risk on delusions. So really, really nice research from the local group. Now some methods of longitudinal analysis. Virtually everything I've talked about so far has been taking a time point and looking at multiple domains and trying to assess interesting patterns at one individual observation. There are different ways of looking across time, which is really important. The most fundamental is a paired t-test. You have before and after groups, and you can evaluate if there's a difference in group averages before and after. And it's not independent groups, it's the same individuals measured at one time point and the next time point. And you're basically just looking at the difference per person. It actually boils down to a single-sided t-test, one sample t-test. Other models that are very popular for looking at longitudinal repeated measures data. One is a linear mixed model, which is a hierarchical model where we no longer just measure group averages altogether, but each person, because they have multiple observations, is essentially modeled as their own. Each person gets their own parameter that kind of describes their trajectory. So if you have a linear mixed model with random effects, you can put a random effect into this model that represents individuals. And because it's a parameterized model and because it's quite a flexible framework, you can add other variables into it very easily. You can look at how individuals vary in their response over time to something if they belong to two different groups. And you can look at interactions between those as well. So, for example, a placebo arm of a trial and an active arm of a trial change in depression scores over time. In the lines represent one individual's trajectory over time. And you can model this at both the subject level and the sample level. And the mixed model does this together. So you end up with estimates for the group average of how everyone performed over time, and then parameters that tell you individual y-intercepts and individual slopes per person to kind of get that sense over time. There are some assumptions in this model which can break down. And again, we'll kind of start thinking person-centric. This offers opportunities for person-centric analyses. There's an extension of this into kind of growth mixture modeling, where instead of assuming that everyone has a kind of different variable slope, which actually follows a deviation that has a normal distribution around the centralized average parameter, maybe there isn't one single population average that people are following. Maybe there are strata of individuals, subgroups of people that tend to follow similar groups of changes over time. And this is a latent method, kind of like PCA was. We come up with a latent class solution that best fit the data. And essentially, in this sense, we can come up with groups of people that have variable trajectories around a group average, but instead of around one group average, it's around multiple group averages. So there's no single sample average, there are multiple averages around which people can vary. And this is kind of the foundation for growth mixture modeling, and it was applied very nicely in this integrative study, where they looked at sex, polygenic risk scores, maternal postnatal depression, partner cruelty, childhood anxiety, 3,500 subjects. Really nice study. What they did was exactly what I mentioned, growth mixture modeling over age, over the course of development. How much do responses on this short mood and feelings questionnaire in these children, between the ages of like 10 and 25, change over time? And they came out with these latent groups. These are the latent classes that they identified, which are kind of sets. These are like averages of groups of underlying trajectories. Childhood persistent, early adult onset, child limited trajectory, adolescent limited, all depending on when, right? At what point in your life do you develop symptoms? Stable low trajectory is a very low response that is stable throughout your transition from childhood to adulthood, right? It's very useful. These now become variables in and of themselves, right? Your group membership to these groups is something that we can then model, and they did that. So they looked at how different types of variables, being female, having a higher low polygenic risk score, having exposures to depression, if your mother was experiencing depression postnatally, how these types of exposures influence your membership to one of these trajectories. And they highlighted a couple of examples of polygenic scores. You can see if you had a high polygenic score, then this is for depression, a high burden of genetic risk for depression, you are more likely to be in the childhood persistent trajectory of depression. And for bullying, you can see that you have a higher risk of being in the childhood limited trajectory and the childhood persistent trajectory. Both of them are higher. And there are interesting ways as to why that might be the case. Bullying, of course, is not a persistent, it could be persistent, but at the time they're kind of thinking that bullying is an exposure that exists at a particular time in life, and then this may influence your likelihood to recover, which is the child-limited trajectory, only experiencing depression in childhood, or childhood persistent trajectory. It's odd that they kind of both came up. But the strongest association by far, this is a logarithmic scale, is childhood-limited trajectory. Now some machine learning. Don't be fooled. I mentioned this yesterday, in a review of machine learning methods for looking at genetic contributions to precision medicine models and treatment response. Linear regression's up there. Machine learning is statistics, and statistics is math. And you can call it whatever you want. You can call it artificial intelligence, machine learning. There's artificial intelligence does mean other things, but linear regression is supervised machine learning. It's a regression problem, it's a minimization problem. So let's just clear that up. So we're still talking about statistical methods. The idea of machine learning is really nebulous. We can talk about that. Here's an example of machine learning applied to actually a precision medicine approach. And I didn't speak about this model yesterday because it's a little more in the integrative space, although not quite as integrative as some other approaches. But I did want to bring it up because it's fairly common. It's a really common study design that you'll see these days, especially because of the availability of packages like scikit-learn in Python and kind of other packages that are becoming available in R to mimic, the early nice functionality of machine learning in Python. It's possible to run many different machine learning algorithms on your data set, kind of in parallel. There's also like superlearner packages where you can string these together. You can string many models together to try to optimize your predictions. It becomes more of like a Lego building. You can put all these pieces together, try them different ways. One of the reasons why machine learning sometimes gets a bad rap, because it's very easy to overfit or derive really individualized solutions to every different problem. So you have to kind of there's a lot of explaining to do as to why this particular thing works for this particular instance of this particular problem. But in this study, they had a sample of about 500 individuals who went through treatment for depression, and they applied linear regression, different types of penalized regression, spline regression, decision trees, support vector machines, and a superlearner method to try and predict who responded and who didn't respond to treatment. And they included a number of different types of variables. They included demographics, they included measurements of anxiety and depression, and also they included some lifetime exposures and medication. What else did they have? I think it says some demographics. But anyway, the variables that were retained in the final model, the best performing model explained about almost 40% of variability in the data set in terms of, which I think their outcome was change in depression scale. So it's not like a classification problem, it's a regression problem. Their elastic net regression performed the best, and it had about 40% variability explained, which is pretty good. And these are the variables that were retained. Really, it's a nice way of going about the study. It's transparent. You have all the data that you collect, push things in, and see what comes out. And then, John, I wonder what machines will do when they learn that machine learning is mostly linear regression. Yes, it is a fact. In fact, even like I'm not going to talk at all about deep learning and like feed-forward neural networks, but if you look at activation functions and how neural networks are formed, it's sometimes blows your mind how simple the individual pieces to these puzzles are and what they boil down to. I'm going to talk a little bit about now tree-based modeling. So this is a little different. A decision tree is simply it's a way of splitting a data set into subsets on decision rules. So and also I should give credit. This I'm using material on the next few slides is from a student of mine, Katrina Quennekin, who's published, it's studies under review, and she put together some of the material on the slides from Dalla, Lana. So you can imagine if you start out with a data set of animals, you can say, does it have 4 legs, yes or no? Okay, well, no, then it's not a dog. I guess some dogs, unfortunately, might lose a leg or two, so this would be an imperfect decision. Does it wag its tail when it's happy? Yes, it's a dog. If not, then it's not a dog or it's an unhappy dog. So you can see it's an easy way to split the data set up and make decisions and classify things. You can see this can be used for classification. It can also be used for regression. But individually, one of these trees is not a very good, we call it a weak learner. And you can kind of see why, because if you have an arbitrary split, again, it's a single split. It's a yes or no question. It's a threshold. With complex data, you're not going to be able to capture the nuances of effects and nonlinear effects in certain types of splits by a single split. So you can do what's called boosting. You can string trees together. Random forest is another way of doing this, but random forest is not boosting. Boosting specifically is fitting a tree and then taking the prediction error from that tree and fitting a new tree to that error. That would be a recipe for overfitting normally, but this XGBoost algorithm, which is called extreme gradient boosting, builds in a regularization step to kind of penalize the inclusion of too many decisions or too much granularity in terms of where you're making your decisions. So you end up with this nice situation where you kind of explain away smaller and smaller bits of error in the model up to a point where you begin overfitting and the model kind of veers away from that path. And then we can interpret these models. Decision trees are kind of nice. You can actually look at it and say, oh, it made these decisions. It's interpretable. I'm going to talk a little bit about SHAP values and what those are and how they work. And SHAP value is just a way of interpreting the model. It gives you get local interpretability. We can look at a single person who went into our model and say, why did this person end up where they ended up? why did they have a prediction of this for our outcome, right? What happened in the model? And also global interpretability. What are the patterns that emerge from the whole model, variable-centric, right? How do features relate to each other? You can also think of local and global interpretability as person and variable-centric in this case. And the way that a SHAP value works is if you think about a model, imagine we have a model here is represented on the bottom layer, a model of any outcome. I guess in this case it's income. The example, and this example is taken from a really excellent article that I've got the citation on the left. It's on Toward Data Science. Best prediction, or the best. Actually, I have this as well. Interpretable Machine Learning by Christoph Molnar. Really excellent reading for anyone who's interested in learning more about this. But also this explanation was not my own. I didn't draw these figures. But you imagine a model of income that includes age, gender, and job, 3 features in the model. You could take those 3 variables and form a number of other models, right? You could have a null model, which is just no predictors, which is just the kind of expected value, which is like an average. Then you can take information of age or gender or job as univariate, right? 1 feature in the model. Or you can have 2 feature models where you combine 2 features, or the 3 feature model. This is like, they call it a power set. It's every possible model you could have as a coalition. This method comes from game theory, so that's the terminology, as a coalition of features. And you can think about marginal contributions. So if you think here, what's the marginal contribution of age in just the model that includes age? So it's like the link, it's the gap between a model that includes no predictors and a model that includes age. Well, the difference is negative 10K, right? If you include age in your model, and this, I should also be very clear, this prediction that's coming out of this is specific to one subject. You see, it's X naught. This is part of the local interpretability. This can be calculated for one individual. So this one individual's prediction, if we didn't take into account any information, would be 50K, but this person's prediction would change to 40K income if we took into account their age. So age is a marginal effect of negative 10K. But our model, our ultimate model, includes age, gender, and job. So how do we understand the marginal contribution of age in all of those scenarios using SHAP? The way that it's done is you take this kind of power set and you weight. You're essentially adding together the marginal contributions of age at every possible permutation, essentially, of this model, of coalition of features, models that includes every possible combination of your variables, and how much is gained or lost when you add or take away age. And the weighting here is what differentiates SHAP from some other types of local interpretability for models. And the weighting just has to do with kind of how much you can learn about a variable from a large model based on how many times it could incorporate into a given model at a certain limited number of features. So if you have a large number of features and age appears in this case, age can appear in 2 possible models that have 2 features. So these weights are both actually downweighted. They don't count as much in terms of what you can learn when you have just this one transition between a model with no features and age on its own. There is more information on the individual contribution of age from this difference than there is from these individual differences. That's part of the theory. So the SHAP for age for this individual in this 3 variable model is negative 11.3K. And I can give you an example of this. This is again studied by Katrina and our group, which is a use of XGBoost and SHAP on a variety of data types coming from a survey of Canadians during COVID-19. There's in our data set, we had 6 time points. These are cross-sectional surveys, not the same people followed up over time. Now there's 7 surveys. 1,000 people responded across Canada at each time point. It was basically from May last year until the end of 2020. And we looked at region, age, gender, lifestyle, cannabis use, alcohol use, home status, like how, do you have children, education, marital status, all these variables, 50 features, and built an XGBoost model. And we applied SHAP to that to try to understand how each variable influences the prediction for each person. And you can visualize them and aggregate them and get different information from it. So in this case, we can look over time of last year, and our outcome of this model, which should be very clear, we had all these input variables. The outcome, the thing we were modeling, was distress. It was an aggregate, actually a latent feature derived from depression and anxiety scores that people responded to. So this is showing SHAP values for basically the contribution that each person had of wave to, and study wave is basically time when we asked them about their depressive symptoms for their wellness, their basically emotional distress. And you can see that levels of emotional distress were predicted to be much higher for most people, and there's variability in terms of the contribution early on, and then over the summer it kind of went down and then went back up, leading to what was the second wave, and into the kind of poorer weather. So we can learn some things about the relationship between time and emotional distress. And then we can also plot each dot here on the right is one individual. And remember, because of this local interpretability of SHAP values, we know how much, for example, the top row is one of our variables. How often do you worry about personal finances due to COVID? And that actually turned out to be the most important feature in our model. And you can see these are the SHAP values on the x-axis, the individual SHAP values, which tell us how the model was nudged, right, for the prediction of emotional distress according to values of this variable for each person. So if you had a high level of worry about personal finances due to COVID, then you actually had the model was being pushed positively. That makes perfect sense, right? You tended to have a higher response for emotional distress. Similarly, if you had a moderate value or not often worried about personal finances, the model was not pushed consistently for this group of individuals in one direction or another, kind of average. And in this sense, you can actually see nonlinear effects for some of these variables. Female gender, yes or no. If you were female, then the model was suggesting you had higher distress than if you were male. You can also look at SHAP values for the intersection of variables, and we did that looking at the interaction of all of the features in our data set, ranked them, and validated with linear regression to put some kind of a value on uncertainty, give it a p-value, and identify this interaction. For example, whether or not you were at high risk for COVID, a perceived high risk for COVID, and whether or not you were hopeful. As the question on the test is, how often do you feel hopeful in the last 7 days? If you felt hopeful all the time, then there was no no difference in how distressed you felt, your depression and anxiety, whether or not you were at risk for COVID. If you were at high risk for COVID, but were rarely hopeful, then your emotional distress was very high. And hopefulness, so in this sense, hopefulness is a mitigating feature. If you are at high risk for COVID, it's better to be hopeful. It's better to be hopeful all around, but specifically if you're rarely hopeful, then being at high risk for COVID would have given you a really high distress level predicted by the model. Finally, high dimensional integration. We've got about 9 minutes left before the break. I think, unless I'm just totally messing up the timing. Someone can tell me if I'm wrong. I thought it was an hour and a half. So I believe we've got about 9 minutes left. High dimensional integration, it's going to lead us into similarity network fusion, which is the method that we're going to do after the break. We're going to go through in R. Different types of integration. This is, again, nomenclature, so you can think about where integration is occurring. Early integration, intermediate integration, and late integration. Early integration is, you can think of it as almost like what happens in a mega-analysis. You harmonize two data sets, combine the data set. You can also perform something like a principal component analysis or a multiple factor analysis, generate a new data set, single data set, perform analysis on that. That is integration. It's done early at the stage of original data. Intermediate integration is something that you see in more like deep learning models that are capable of taking multiple inputs of different structures and different sources and analyzing them together in one kind of framework that often combines and reduces them into the results, the outputs, and the predictions. Late integration is more along the lines of what we see, kind of more often what we see like meta-analysis, where you have each data set separately or data type analyzed on their own within their own frameworks, generate a set of predictions, and then integrate those predictions at the kind of last step, at the late integration. And what we're going to do today is more of kind of early integration step. So similarity network fusion. This is one of a large set that I'll show at the end of data sets that have emerged in the last 5 years, no longer, to cluster individuals, take a subject-centered approach, and identify the similarity of people based on huge numbers of variables. So with omics technologies, RNA sequencing, DNA methylation, histone acetylation, ATAC-seq for chromatin remodeling, you have hundreds of thousands or millions of data points per person. How can you start to understand who's similar to each other, and what types of non-traditional statistical methods can be developed in order to do that? The idea behind similarity network fusion is that each data type has its own kind of, so each data set has its own kind of, this is a mixture of early and late. We can talk about it, early and late integration. Each data set is subject by variables, right? And there's a way to identify the correlation between variables, as I mentioned before, or if you transpose that matrix, the similarity between people. You can identify these affinity matrices, which basically shows how similar each person is, and you can define those affinity matrices separately for each of your data type inputs. So I might have gene expression, I might have methylation, I might have some imaging variables like from EEG, I might have cognitive variables from a set of cognitive tests. I can understand how similar each person is within each of those domains separately, right? That matrix can be represented as a network, right? You could picture each person as a dot, a node. How similar is each person? If they're similar enough, they're linked to each other, right? You can imagine how people might cluster. And then the final step is this fusion step, which is what similarity network fusion does. Because we can identify subtypes according to one modality or another, just with that first step, these affinity matrices, what happens when we fuse the networks, when we use information? Because now remember, if you have the same set of subjects in each of your data types, the affinity matrix is going to be a matrix of subjects by subjects, right? Pairwise. How similar is each person to each other person? Those matrices will have the same dimensions, right? It's going to be all of the people in your study, how similar are they according to one data type, two data types, three data types. Use information from each of those similarity matrices to update each other. It reinforces the similarities that exist in multiple data types, and it begins to degrade similarities that might be specific to a single data type. And so this is what it looks like. We have variables and subjects, but we're not looking at variables in columns, we're looking in rows, at individuals. We can take this, we choose a distance metric. This is a point we can talk about as well. Certain distance metrics work for large numbers of variables and some don't. But again, we've turned a matrix of 30 subjects in this example by 7 variables into a matrix of 30 subjects by 30 subjects, right? It's just telling us how similar are each person if we correlate people across variables. And then we can define the distance as just how different people are. We can define the affinity or similarity of that to get these kind of similarity measurements to kind of identify subgroups of people who might cluster together. This is just a graphical example of what I'm talking about from the actual methods paper, and you can see here they've used mRNA's kind of RNA-seq data and DNA methylation as the examples of 2 molecular data types that are fused. So you have your original data, you get these patient similarity matrices, and then the iterative fusion step updates over multiple steps the networks until they converge. They converge onto a unique solution. And this is an example from simulated data. If you imagine a ground truth where you have 2 people who are 2 groups of individuals, precision medicine, you say two sets of people, blue and red, who are quite different from each other according to these 2 data types, and then swap out some for the other, right? Make it an imperfect grouping. So take some of these individuals and have them look more like blue, and then take some of the blue people and have them look more like red. If you merge those people together and create these fraudulent data types where the groups aren't totally separated, so let's say data type 1 is methylation and data type 2 is gene expression, you have these really imperfect clusterings according to the ground truth for each data set separately, but after fusion you actually get a much cleaner separation between the 2 and a ground truth, and it outperforms other methods that have been developed prior to SNF. They applied it to breast cancer. They found compelling findings, of course, in their study. They would have to find compelling findings. It's been applied by groups at CAMH. This is Laura Stefanik under the supervision of Aristotle Vaneskos, who did a study applying similarity network fusion to data from looking at multiple disorders. This is healthy controls, bipolar disorder, autism spectrum, and schizophrenia spectrum. And the input data for this, I believe, so a while ago, there was some imaging data. I think it was diffusion imaging data, demographic data, and cognitive data. And so taking these three kind of data types and fusing them into a network revealed 4 subgroups of individuals that did not map perfectly onto established DSM-5 diagnostic criteria. So this is kind of the value of using a data-driven approach that includes some biology, some objective cognition, some demographic information to kind of break the mold of traditional diagnoses. And the idea is that these groups may have more homogeneous properties, kind of biological and psychosocial properties that are more similar to each other and may therefore experience similar trajectories of symptoms or response rates to medication better than the traditional diagnosis, which, as we heard about on day 1, you can have very different symptom profiles and different underlying biology and still be grouped into the same diagnosis. So it's an example of that right here. This is just for due diligence, probably useful because you can't read any of it. SNF is in the middle here. This is a very recent review, a list of multi-omic clustering methods. Very new. A lot of these have come out. Late integration, early integration, dimensionality reduction, and then similarity-based methods, which are kind of person-centric methods, spectral clustering, SNF, and a few others that I'm not familiar with. But there are options out there. So that's it. The workshop, after the break, when we come back, we're going to do SNF. We're going to run a similarity network fusion on some simulated data. So I've got some data for EEG recordings, data from methylation profiles, and also data from gene expression. So we're going to have 3 different data types. We're going to fuse them into a network. We're going to try a couple of different clustering solutions to identify subtypes, and then kind of look and reflect and see what do these subtypes map onto in terms of withheld data. What's the use of them? What is the difference between the subgroups? That's what we're going to do after the break. You can go to the summer school GitHub and follow along if you'd like. The Docker has been set up. The binder has been set up. It seems to be functional from what I'm aware of. And it's just an R Markdown script. It's not a lot that you need for this. The Docker has everything in it. It'll all be in RStudio. So we'll come back after the break and we'll get right to that. Just want to verify timing. So can I get a thumbs up from somebody? We're going to come back at 10:45. Did I go 15 minutes over? I thought it was 9 to 10:30 and then 10:45 on, but I probably blew it. Probably messed it up. Thanks for the claps. Are we going to do 10:40? We're going to do 10:45 to noon. I think that's the plan. I'm just going to make it up. Someone can tell me. Aaron can tell me, perhaps. But anyway, that's my plan. So thank you for listening. I hope you got some perspective on integration. Thanks, Aaron. Thanks, Marta. Yeah, perspective on integrative methods. We covered a lot of ground, a lot of very different types of study designs and ground. I hope it was informative. Hope maybe you got a method or an idea out of it. And again, I really want to emphasize, write down your questions on your paper, on your whatever, on the Slack channel of questions for the afternoon. We're here for one day only, right? We're only here for one day. This is your chance to ask us the questions, not just about science. I should also say questions about who we are, where we came from, career advice, if that's the kind of thing that you're into. Any thoughts you have about your training or about decisions you make. We're open for that. One day only. We're not going to try to fill a whole afternoon of chatter. We're going to answer the questions and kind of go from there. So my advice, take the opportunity. I'll see you back here in 15 minutes, and we'll do some similarity network fusion, and tell your friends. Thanks, everybody.